Hello, hello.
Hi, Jeff. This meeting is being recorded. This is Bootcamp number 4, and we're talking today about the Hypothesis Center, which has been developed by dkNET. And also, we're going to be speaking with Dr. Neil McKenna, who has developed the Signaling Pathways Project, which is an amazing new tool, a data-driven tool that is going to be used by the D Challenge. So I'm going to introduce Dr. Jeffrey Greth coming to us first from UCSD, and he's going to just introduce us to the Hypothesis Center and talk about what it is and why it's useful and helpful. Thank you so much for joining us.
Thanks, Monica. So what I'm going to do, since many of you, this might be your first boot camp, I'm going to give a 1 or 2 slides about dkNET itself to give an overview, and then talk about, as Monica mentioned, the Hypothesis Center, where we are aggregating information about bioinformatics tools and also providing guidance on how to use those tools. And then I'll hand off to Dr. Neil McKenna from Baylor College of Medicine, who has developed the Signaling Pathways Project and provides the ability for bench researchers to actually get in and really explore and dive deeply into omics data through user interface. And so dkNET itself, was funded by the National Institute of Diabetes and Digestive and Kidney Diseases to provide an information portal for biomedical researchers. Really what we're trying to do is put researchers in contact, with information about resources, help them with, for example, mandates around fair data and making data fair. Also discovering resources and data and information that might be available to them. So there are sort of these 5 key areas in the portal. The top 2 really focus on resources, discovering resources that may be useful, but also meeting the mandates from NIH around authentication of resources such as antibodies. There was a boot camp that covered this on August 20th, so please go back to the video. There's a link here where you can get to all the boot camp videos. As I mentioned, services around fair data. There was also a boot camp on June 22nd that you can go back and take a look at. And what we're talking about today is the Hypothesis Center, which is, again, one of these key areas. There was a boot camp sort of giving an overview of all these areas from August 6th, and so you can also go view that. And so, as I mentioned, there are a number of tools that dkNET provides, and these sort of span the research life cycle. Everything from finding information, finding data, finding grant funding opportunities through the discovery portal. You can then use the Hypothesis Center to really help formulate potential hypothesis for your experiment. When you're planning your experiments, again, these resource reports, authentication reports, will be critical. When you actually start analyzing your data, finding the right tools for analysis, but also digging again into the Hypothesis Center to explore, what your data might mean. And then publishing your results includes also publishing your data. And so this is where the fair data resources come in, and also ensuring that you are properly identifying the resources such as antibodies, cell lines, software tools that you used in your research study through the use of research resource identifiers. And so again, we're trying to provide, information, to assist researchers, in doing their science. And, what we're, again, focusing on today is the Hypothesis Center. So really, an area of the dkNET portal where, we allow users and we point users to tools to allow them to explore diverse data, and generate research hypotheses. So if you follow the link to the Hypothesis Center in this bottom right of the quadrant on the main page, you'll end up on the Hypothesis Center landing page, and there are a couple areas here that are of interest. One of them on the bottom is that, as we bring in additional tools and, there is additional research being done, we are making these stories of discovery available. And so these stories are, linked to research papers that have been developed using the tools that are outlined in the Hypothesis Center. The top left is the getting started area. This is where you start by selecting what you actually want to explore. And so under the get started, there's a drop-down menu, and that will touch on that on the next slide. And then on the right, we have sort of the feature tutorial, but we also have new tutorials that are being developed. So in addition to the signaling pathways project and the mouse metabolic phenotyping center that we have initially integrated, we're also working with Avi Mayan's group to integrate tutorials around his Apiter tools. We've also been working with the Type 1 Diabetes Knowledge Portal to pull in tutorials there, and then also the Hearn Resource Center is also providing some tutorials for us. So again, this is an area that will be expanding and continue to expand over the next year as we bring in more partners and we bring in more tutorials to sort of build out what we are making available. So to get started with your tutorial, in the top left box under Get Started, what you do is you can select one of the questions that is of interest to you. So here, we're looking for evidence for target genes of a signaling node of interest. And then there are a couple potential tutorials that are available that tie into specific data in that tool. And so basically what we're doing is we're providing a couple sets of tutorial information about what data and what information you need to put in the tool to guide you through that tutorial. And so to get started, then you just click on the blue tutorial button and you'll end up on the tutorial overview page itself. And so here, the first thing that you're seeing is the introduction to the tutorial, and also in the right hand side, if we have any prior webinars or tutorial videos that are available through our YouTube channel or other YouTube channels, we make that available as well as part of this tutorial page. As you then go down the tutorial page itself, the first thing that you'll see is some information about what you'll learn in this tutorial, so getting you oriented. And then we will jump right into the lesson itself. On the left of the page in these boxes are the textual descriptions of the step in the tutorial that you are currently on. And on the right are either action buttons. So for example, in step one, we launched the signaling pathways project for you by clicking the green button. And then in step two, you can see that we're outlining on the SPP site, where you need to click to move on in the tutorial. The other thing that we provide in terms of the tutorials is we go beyond just outlining the steps that are required. We also try to provide the scientific rationale and a bit of scientific information about the steps that you are going through and will be going through. And so this is something, to provide additional information that as you go through this tutorial, you're not just learning how to click the buttons, but hopefully you're learning a little bit about why you're clicking the buttons that you are. At the end of the tutorial, once you've gone through all the steps, we also provide an overview of interpreting the results of a tool that is documented with these tutorials. So give you a little guide, as to, what you're seeing, in the results and how you can interpret that. So we try to really provide sort of beginning to end instruction on the tools themselves. And then in addition to the tutorials around these tools, we do have information pages for each tool that we provide information on in the Hypothesis Center. So this is the Signaling Pathways Project page within the Hypothesis Center. And it provides, again, a number of different areas. So if you look, on the top of the page is, again, information about the tool. On the bottom, you'll see that there are quick jump points, so if you're looking for data sets for SPP, there's a link, and then there's, a set of other links below that in terms of finding, specific example use cases that you can click through to. And then on the right, there are also a set of further on videos, webinars that you can link to. So again, trying to provide an overview of that resource that is being provided. And so now what I'm going to do is I am going to hand it off to Dr. McKenna. So let me stop my share real quick and make him the host. I will make you the host, Neil. Okay, so Neil, you're now the host, so you should be able to enable your video. Okay.
Okay.
Thanks so much for that.
Yes, thank you, Jeff.
So welcome, Dr. Neil McKenna from Baylor University in Texas, and he has created.
It's a Baylor.
I'm sorry, Baylor College of Medicine in Houston, Texas. Sorry, but he is the sort of the really creative force behind the Signaling Pathways Project, which is just an amazing effort. It was used in the COVID setting, and we're using it now in the D challenge and the type 1 diabetes setting, and I can't wait to hear what he's going to present. So thanks so much for joining us.
Thank you, Monica. Okay, so the research. Sorry. Just bear with me for one moment, please. So the work that I'll be, discussing this afternoon is shown in 4 of our recently published articles. And I will show this slide again at the end of the seminar. So I want to walk you through 3 real-world applications of SPP analytics that will form part of the kind of informatic engine that will be the initial version of the Hypothesis Center. And the emphasis here will be on SPP's investment on making full use of existing resources that enables investigators to add value for their own research. So I suppose we should start by explaining what we here at SPP understand as signaling pathways. So the term signaling pathways, as we understand it, refers to a series of functional and physical interactions that actuate the cellular response to a signaling event. And this signaling event may be a hormone or some other signaling molecule. It may be an environmental substance, or an interaction in the extracellular matrix. It's really any event or the surface or even inside the cell that involves a cellular response will actuate a signaling pathway. And cells respond to signals in many ways, but arguably the most important of these are those that occur in the cell nucleus. And specifically, those whose endpoint is increased expression or decreased expression of a particular gene. And you're likely, familiar with this kind of, a depiction of a signaling pathway. Maybe it's a figure in review. And what these figures almost invariably focus mostly on are what we refer to as the nodes in these pathways, which are the molecules whose physical and functional interactions transmit the signal from the membrane into the cell of nucleus. And the three major signaling nodes encompass receptors, which initiate the signal, enzymes, which modulate the signal in some way, and then influence the function of the third type of signaling node, which are the transcription factors. But what's often marginalized in figures like these is the impact of these signaling pathways on expression in the nucleus. There's maybe some kind of vague allusion to some kind of downstream process, but often there's very little in the way of the detail in which the rest of these pathways are shown. So most of our knowledge of cell signaling is obtained from the research literature in the form of research articles or in reviews, which aggregate facts from multiple research articles into a single article. However, there's another universe, if you will, of information that informs upon cell signaling in the form of archived omics experiments. And again, for the purposes of SPP, we're going to focus on a couple of these, and those are transcriptomics, which is represented by expression array and RNA-seq, and then also cystomics, which is represented mostly by ChIP-seq experiments. Now, even though omics platforms are designed to inform upon thousands of events within a cell, as a consequence of the way that research is done, what often happens is that in order to get an article published, investigators are forced to kind of focus on a small handful of those events, which are then looked at in more detail. And so what happens is that there's a lot of information that never makes its way into the research article in any level of detail. And even that information, it wasn't of immediate relevance to the investigators, it may still be useful for other researchers. So the goal of SPP is to reuse existing archived omics data sets to identify high confidence transcriptional targets of signaling nodes in the form of what we refer to as our consensomes. And what consensome analysis does is to survey across independent archived data sets that involve a common molecular perturbation, and to rank the underlying targets by the frequency with which they are differentially expressed across those independent data sets. And we can do something similar for ChIP-seq data sets. When you hear the ranking is based upon the average peak height for a specific target across independent data sets that have a common IP antigen. And so I'll use the example here of the insulin receptor family, Consensome. And this is based upon numerous independent experiments that all have one thing in common. And that is that they involve some kind of functional perturbation of a member of the human insulin receptor family. And we can use the resulting ranking to identify transcripts that have consensome values of less than 0.05. We refer to these as confidence transcriptional targets, or CTs. And those in the 95th percentile of the consensome as high confidence targets, or HCTs. So the first story of discovery, if you will, is one that used our Fair-Omics platform to establish consensus around the human transcriptional response to SARS-CoV-2 infection. And this is work that was done with my longtime colleague here at BCM, Scott Oxner, and also at Rudy Pillick at the Idegger group in UCSD. And specifically, we wanted to identify a reduced bias approach to identifying those targets whose differential expression is most consistently involved in response to SARS-CoV-2 infection, both at the level of expression, but also to identify nodes that are involved in the response. And in order to do this, we applied consensome analysis. But here we weren't computing node consensomes, but rather consensomes for SARS-CoV-2 infection. And so we surveyed across numerous independent experiments where the investigators had infected cells with SARS-CoV-2. And the results are depicted here in the familiar form of a volcano plot, log SC on the x-axis, and on the y-axis, the minus of log 10 of the consensome Q value. The first thing that we wanted to do was to validate the consensome, and in order to kind of convince ourselves, but more importantly, other people, that it was a a reliable resource. And we can kind of eyeball the consensome and identify the sort of usual suspects, if you will, of the innate immune response, such as the various members of the various interferon-induced families. But in order to rigorously validate the consensome, we needed an independent benchmark that was focused specifically on SARS-CoV-2 or COVID-19. And one such study appeared earlier last year in the form of a Nature paper that identified a signature of 9 targets, SNPs in which were overrepresented in critically ill COVID-19 subjects. And consistent with the reliability of the SARS-CoV-2 consensome as an environment for modeling the transcriptional impact of SARS-CoV-2 infection, the distribution of these 9 targets was heavily skewed in the direction of SARS to HCTs. So, having established that we were able to use consensome analysis to recapitulate the transcriptional impact of SARS to infection, we next wanted to go one level up, if you will, and to see if we could adopt an omics-based approach identifying nodes that were involved in orchestrating these expression events in response to SARS-CoV-2 infection. And in order to do this, we developed a new analytical pipeline, HCT intersection analysis, which, as the name implies, computed intersections between SARS-2 HCTs and those from our library of nodes. So HCTs that we had identified in our node consensomes. And in the next few slides, I'll be showing these intersections in the form of a scatter plot of the nodes, in which the odds ratio of the over the expected intersection is left to right on the x-axis, and the confidence of the intersection increases from the bottom to the top on the y-axis. And when we ran HCT intersection analysis on the SARS-2 Consensome HCTs, we saw a robust HCT footprints, if you will, for nodes with known roles in the innate response. In the innate immune response, such as IRF1 and STAT2. But again, as with the Consensome, we needed an independent SARS-2 focus study against which we could validate the nodes that we were predicting were involved in this cellular response to SARS-2 infection. And in order to do this, we sourced a genome-wide CRISPR screen for factors that had important roles in the host response to SARS-CoV-2 infection. And again, reflecting the reliability of our predictions, nodes that we had predicted had roles in response to SARS-CoV-2 infection were overrepresented in the most highly ranked hits from this screen. Now, as an example of a use case that can be gleaned from our analysis, we identified one of the nodes that was highly activated in response to SARS-CoV-2 infection was the progesterone receptor. And it's well known that loss of function of the PR in other organs is involved in increased expression of these interferon regulated targets. And so from this, we hypothesized that PR agonists might be used to treat COVID-19. And indeed, there was a paper published earlier this year that showed that progesterone was actually effective in the treatment of males hospitalized with COVID-19. So in order to make this universe of the data points available to the community, we entered a partnership with the index resource, in which we were able to render these networks in the form of sort of a familiar stethoscope networks. And these enable the researchers to interact with these networks in a very engaging way, and also to link into SPP for the actual underlying experiments. And you can read more about this in our recent article. So the second story of discovery is a collaboration with Dr. Karl Dimitar-Bissek of Duke University School of Medicine. Dr. Bissig's group has developed a humanized model of NAFLD in which human and mouse hepatocytes exist side by side in the mouse liver. In response to high-fat diet, the human hepatocytes accumulate neutral lipids in the form of cholesterol esters, whereas the mouse hepatocytes, did not. And so the goal for SPP in this collaborate was to validate this model against transcriptional networks that are involved in clinical NAFLD. And so we focused on a set of 12 transcripts that were differentially expressed in response to Western diet, both the human and mouse hepatocytes, and whose relative expression levels were at least 1.2-fold higher in the human hepatocytes relative to the mouse ones. And remarkably, and consistent with the histological analyses, 6 of these transcripts, half of them, encoded enzymes which are in the novel cholesterol biosynthesis pathway. And we refer to these as CDP-EGs. Now, this was interesting, but was this an accurate recapitulation of clinical NAFLD? And so, in order to answer this question, we again turn to Consensome analysis and computed a NAFLD consensome across 22 independent expression profiling datasets that compared expression in clinical NAFLD and normal liver. Now, I mentioned earlier the importance of a validation of our analytics, and so in order to do this, we benchmarked the consensome against a 25 gene signature recently shown to correlate with the severity of clinical NAFLD, and we showed that these transcripts are actually robustly overrepresented among NAFLD-induced HCTs. And confirming the clinical relevance of the model, 4 of the enzymes that I referred to earlier are actually in these NAFLD HCTs, and these are heavily overrepresented here. So what we wanted to show also was whether the model accurately identified transcriptional drivers or nodes that are regulating the expression of targets in NAFLD. And so to refresh your memory of these intersection plots that I'll be showing in the next couple of slides, the position of the node in this slide indicates from bottom to top increasing confidence on the y-axis, and from left to right, increasing odds ratio. So again, in keeping with the theme of validation of existing benchmarks, we sourced a study that identified a set of 5 nodes, motifs for which were enriched among targets that were associated with the incidence of NAFLD. And I'll refer to this set of 5 nodes by the author of the first author on the study at NAMJU. Now, when we performed HCT intersection analysis on the NAFLD CTs, we found that the NAMJU nodes were heavily, very heavily overrepresented among nodes that had high rankings in our analysis. So we next performed HCT intersection analysis, and we find significant intersections for 4 nodes, 2 of which are members of the SREB family, which have well-known links to hepatic steatosis, and a couple more that had been previously linked to dysregulation of lipid metabolism. And to underscore the relevance of our model to clinical NAFLD, the distribution of these nodes skewed heavily in the direction of nodes that had high rankings. So my last story of a discovery relates to a collaboration with Olivia Osborne of UCSD, and this it actually involved work with a summer student who was a dkNET intern last year. And one of Olivia's areas of interest centers around mechanism underlying antipsychotic induced weight gain, with the goal of understanding why some individuals would gain weight in response to antipsychotics while others won't. And so in order to study this, Olivia's group set up a mouse model in which your baseline blood was taken from mice who were then given the antipsychotic olanzapine for 2 weeks, and then they split these mice into mice that gained weight in response to olanzapine, and those that did not. And so they identified 2 sets of baseline PBMC transcripts, and those were at least 1.5-fold higher in the weight gain of prone mice, and those are referred to as AI at WGP, and those that were 1.5-fold higher in the weight gain resistant mice over the prone, and these are AI-WGR. And so, introducing SPP again, our goal here was to validate the model in the context of mice, mice metabolism, and also clinical antipsychotic induced weight gain. And we hypothesized that AI-WGP would be enriched for genes whose disruption in mice would result in increased weight gain. And in order to answer this, we used a couple of resources, MMPC and IMPCC, which map genes to physiological functions. And so from MMPC and IMPC, we retrieved 2 sets of genes. Those whose knockout in mice resulted in decreased total fat mass or slow postnatal weight gain. And our rationale was that transcriptional drivers of AI-WGP genes would be enriched within these sets of targets. And we find that indeed AI-WGP nodes, when we looked at the AI-WGP nodes, the nodes that are encoded by these weight gain targets were overrepresented among AI-WGP nodes. And finally, we wanted to put all this work in the context of human weight gain in response to antipsychotics. And so we used an existing study that showed that 155 human genes whose blood expression levels were associated with clinical body mass index. And consistent with the clinical relevance of the model, mice orthologs of the clinical BMI associated genes were distributed almost exclusively in the eye of WTP targets. Now, as you are aware, SPP is involved in the dkNET Sugar Science Deep Challenge. And as part of our contribution to this, we are organizing ARCAD experiments where investigators have compared expression profiles in various types of blood cells between individuals with a type 1 diabetes and healthy controls. And the aim of this is to enable de-challenge researchers to easily or more easily formulate hypotheses around novel signaling events in blood cells that may be involved in type 1 diabetes. And one of the products of this work will be the T1D Consensome, which consistent with the other consensomes that I discussed. This ranks human genes according to the frequency of their significant differential expression across all of the underlying type 1 diabetes versus control experiments. Now, since I alluded to earlier, this is a very a diverse set of experiments. So in addition to the consensome, we will be giving investigators all of the underlying differentially expressed targets from the individual experiments. And so the goal here is to give investigators a kind of a high level view of what's happening. So again, in our partnership with the IDIGER group at UCSD, we will be making all of these networks available through their index resource. And just as a quick example of the insights that can arise through the consensome analysis, you can see here that one of the most frequently differentially expressed genes between type 1 diabetes blood cells is TNF. TNF, of course, has a historically very well documented connection to type 1 diabetes, and the anti-TNF antibody belimumab has been recently shown to be effective in new onset type 1 diabetes. However, there are other targets of ligands in this case that have not been previously associated with T1D that the consensome shows are actually very frequently differentially expressed. And so these represent potential previously unexplored opportunities for your intervention in the disease. And we will be releasing the consensome and all the other associated networks for the D-Challenge at the end of the month. And with that, I'll take any questions.
Well, this was an excellent presentation. Really just a great walkthrough about the power of the consensome, the power of what signaling pathways is doing, the new way of mining data, the new way of representing it, and sort of, I loved how you said in the beginning, you're going back to sort of like look at what was sort of, cast aside. It, wasn't used by the researchers, but it still can be quite valuable. And I loved that you also went through and talked about, how this was really used just recently in the COVID, circumstance in the COVID realm. And I mean, that's very powerful. that you guys were able to go in and collect these data, reimagine them, not really even sort of creating a new way to look at that data. So it's just fantastic. And I'm hoping some folks from the audience will just send something in the chat regarding questions. Oh, here's a good question. Great talk. Thank you. In type 1 diabetes, given the disease pathogenesis, heterogeneity, and exposure to differential environment, what do you think about the power and sample size for the GE analysis? Does this study also involve samples from family and siblings? Great questions. Thank you.
Yeah, so, these are good points. Regarding, the power and the sample size, when you're in the kind of fair data realm, you kind of have to work with what you have, right? So, in an ideal world, we would have a very large number of archived experiments or archived data sets that would kind of enhance our statistical power. Unfortunately, it's really only recently that authors have been actually, as part of the kind of a publication process, they actually have to archive their a data set. So when we were actually in the process of kind of building this Consensome, we actually found a lot of interesting articles that where investigators had used expression profiling, but unfortunately they hadn't archived their data sets. And we can't, in order for the Consensome to be as reduced bias as possible, we have to use archived data sets where they have looked over a large number of data sets, because as I mentioned earlier, typically what investigators include in their supplementary data are only those targets that are actually differentially expressed. But we also want to know which ones aren't, right? Because then you can get an idea of the significance for a particular target for over the kind of underlying experiments. And to your last question, does this study also involve samples from family and siblings? I believe it does. We are going to along with the Consensome, we will be providing as much detailed information, metadata from the original study that we were able to find. And if there are particular studies that do focus on families and siblings, then that information will be in there.
That's a great answer. I had another question here. How easy will the D-Challenge be to use, or the resources to use for those who are sort of novices in bioinformatics?
That's a good question. So in addition to the, visual interface through, Cytoscape, oops. You'll notice here that we have for each target, you can click on it, and that will pull up a window that will show you a list of contextual links to related resources. There are links into SPP, which will show you various signaling nodes that are involved in the regulation of that target. There will be a link into dkNET, where the investigator will be able to find resources, resources, reagents, antibodies, etc., that are specific for that particular target. And in addition to this, Cytoscape interface, we will be providing the consensomes and the other networks in the easily accessible form of Excel, spreadsheets, so that even using a very simple stats, very simple statistical approaches, that anybody will be able to identify whether their particular targets of interest are significantly differentially expressed between individuals with type 1 diabetes and abnormal individuals.
That's great.
And I just want to follow up real quick. I mean, that's the other reason, from the beginning of the talk, why, we're also developing the Hypothesis Center. Really, it's to enable the use of these bioinformatics tools by novices, by bench researchers who may not be bioinformaticists, right? So what we're trying to do is provide, the appropriate materials and information through these tutorials to get people, make it accessible to them to use those tools.
Yeah, and I also think what the power of this is many different scientists can come in from many different avenues of inquiry. So for instance, if you're interested in membrane proteins, if you're interested in pseudogenes, transporters, whatever, you can interact with this consensome that Neil and his team are building and sort of dive into your world, your interests. It's in this way, it's very diverse in terms of what it's representing, but it's also catered and curated to your interests. Which I think is great.
Yeah, and Monica, you actually mentioned pseudogenes here, and this is actually interesting because we've done a bunch of different consensomes now for disease states. And this one is actually unique in the respect of the number of different pseudogenes that are actually differentially expressed. As this name might imply that these aren't worth thinking about pseudo, right? But there's actually a lot of evidence out there that shows that these things are actually functional within the cell. So you have things like this pseudogene here, TP2, P8, these other ones here that as this kind of deep red implies, these are actually pretty frequently differentially expressed. So this is an environment through which we can identify events like that and make them a lot more accessible for researchers.
Yeah, agreed. I think it's really in some ways this is a gold mine of resources and it's an easily accessible one. So I think that we would invite people to just, take a walk through what it is we're offering and maybe even, sign up, come to the very first meeting. There's going to be a lot of support for people to move through these tools and just sort of explore it and be open to it. It's a new way of doing things, and we're all about that, doing things in a new way. We are very much involved in supporting the dkNET movement, where they are trying to get more open science, more shared data, more fair data. So there will be more archived experiments and data sets, as Neil referred to earlier. And this Hypothesis Center, wedded together with the Consensome, is a very powerful onboarding for all kinds of scientists to try to, enter and engage and explore in this new data set. So thank you all very much for this excellent presentation. It was superb. And we hope to see everyone at, registering at the D Challenge and joining us on October 1st for the very first meetup and, introduction to what it is they'll be engaging in. Thank you again.
And there was one last question here. I kind of have to.
Oh yes, sure. Sorry, I didn't see this. Yeah, I just saw a new one.
Yeah, anybody can email me at mckenna at bcm .edu.
Yeah, and Neil will be available sort of as a guide for scientists throughout the process, mckenna at bcm.edu. And you can have some more in-depth conversations with him about the nitty-gritty details. Thanks for everyone for attending, and thanks for our fantastic panelists.
Okay, thanks everybody.
Have a great rest of the day.